Okay, everybody, welcome to Chapter 4, where we're going to dip a little bit into cell biology and chemistry, which are important to get a background in order to understand what we're going to discuss in microbiology. We'll talk about the composition of cells, we'll talk about the foundation of biochemistry and chemistry, talk about relevant biological molecules and their role in the life process, describe how cell membranes work and what transport mechanisms they use in order to get the materials they need and dispose of the wastes that would threaten their existence. And we'll talk about examples of how pathogens take advantage of these processes. In this section, we'll talk about how atoms form the molecules of life and describe the kinds of bonds atoms form to make molecules and explain the properties of water. Okay? So, in order to begin this discussion, we have to start all the way back at the creation of the universe. Okay? At some point in the distant past, everything in the universe was concentrated into a tiny little single point, which was what physicists call the singularity, and that was an unstable situation. And that led to a gigantic explosion known as the Big Bang. And when that explosion took place, everything in the universe was created. And the universe is made up of three things: matter, energy, and empty space. Okay? Matter is anything that has mass and occupies space, and it comes in three flavors: solid, liquid, and gas. Okay? Energy is the ability to do work, and empty space is empty space. Okay? There are two laws that govern the entire universe. Okay? They're called the first and second laws of thermodynamics. The first law of thermodynamics states that energy and matter can't be destroyed or created. And the second law of thermodynamics states that if you consider any system, a system could be a glass of water, or the air in a room, or it could be the dust on the ground. Okay? If you don't put energy into that system will find the lowest energy state, meaning it will arrange itself in a way that takes the least amount of energy to maintain. Another way of thinking about the second law is that basically things take the easy way out. Okay? This, for instance, is why water runs downhill, why cars don't roll up hills all by themselves. Okay? This is why smoke rings diffuse in a room. All right? All of those are processes that tend towards the lowest energy state. All right? The three forms of matter, solid, liquid, and gas, have different properties, right? A solid maintains its shape and its volume, and the components of the solid don't move very much. They kind of vibrate at fixed points. Okay? A liquid is made up of components that have a little bit of movement to them. All right? So the components in the liquid can slide past each other and around each other, and that's why a liquid forms to the shape of its container. And then a gas has even more movement associated with it, such that a gas will touch all the surfaces of the container that it's in, like the air in the room you're in right now. Okay? You can shift from a solid to a liquid, from a liquid to a gas, and in other directions by changing conditions of temperature and pressure. Okay? Any matter that you can think of has the ability to exist in any of those states. Okay? The atom is the smallest unit of matter. All right? And it comes from an ancient Greek concept. A gentleman named Democritus in ancient Greece was staring at a boulder one day, and he thought that if I took that boulder and I busted it in two, I'd have two smaller boulders that still had the property of the original, and if I did that again, I'd have four even smaller boulders that still had the properties of the original. But I would eventually get to a point, if I continued this experiment, where I wouldn't be able to divide that particle any further. And he called that tiniest piece of matter the atom. from the Greek a, not, and tomos, divisible. Okay? And that's basically the concept of the atom we have today. The atom is the smallest unit of matter that still has the chemical properties of a larger quantity of that same element. Okay? And what are elements? Pure forms of matter made of atoms of a single type. Okay? And surprisingly, there aren't that many elements in the universe. And I'll show you later um, what the elements are and where they're at. Okay? Molecules are combinations of atoms. A compound has at least two different elements in it. An organic compound contains at least one carbon and a hydrogen bound to it. Okay? And the reason they get the name organic is because organic compounds are usually associated with life. And biomolecules are organic compounds that are found in cells and organisms, bio meaning life. Okay? The way that the elements attach to each other, all right, is through the production of what are called chemical bonds. And we'll find out that within a molecule there are two kinds of chemical bonds that we can describe. There are ionic bonds and there are covalent bonds. Okay? You can see some examples of different elements in living cells here. We usually designate the symbol for the element with a capital letter and maybe in some cases a capital letter and a lowercase letter, as you can see down here with sodium, right? And then you can see a property known as the atomic number, right? And then another property known as the atomic mass, okay? So we're going to find out that atoms are built from particles that exist in different locations. Particles called protons have a positive electrical charge and they exist in the nucleus of the cell. The same with a particle called the neutron, except that it has no electrical charge. And then the electrons have a negative electrical charge and are found at locations outside the nucleus that are called orbitals. Okay? The atomic number defines the identity of the element because the number of protons and the number of electrons in an element are always the same. So every element is electrically neutral. That means that the chemical properties of the element are defined by the atomic number. And the chemical properties are basically how the element reacts with other elements, right? Does it form an ionic bond or a covalent bond or a metallic bond, depending on what other element it interacts with, okay? And when you get down to it, really that's all that chemistry is about, is the making and breaking of chemical bonds, okay? You can see some numbers over here on the right, bonds shared in molecules, right? Hydrogen can form a single, what we call a covalent bond. Carbon can form 4, nitrogen 3, oxygen 2, sulfur 2, phosphorus up to 5, okay? And sodium generally forms an ionic bond when it reacts with a nonmetal, or a metallic bond if it reacts with another metal, all right? So what we should do now is take a look at sort of a description of all the elements in the universe. Okay, so when the universe was created, all the elements that made up the matter in the universe were created as well. And believe it or not, these are all the elements in the universe. And when you consider that the universe is infinitely large, it's pretty amazing that there are so few elements actually in it. Okay? You'll notice that this chart here, called the periodic table, is arranged in rows and in columns. Okay? The columns are called groups. Group 1A, 2A, 3A, 4A, 5A, group 5B, and so on. Okay? And the rows are called periods. Okay? The mass of an atom of an element gets heavier from top to bottom and from left to right. Okay? So the heaviest stuff is in the lower right-hand corner of the table, and the lightest stuff is in the upper left-hand corner of the table. Okay? You'll notice some different colors on the chart too, okay? This little staircase here, all right, is the dividing line between the nonmetals here, kind of in silvery gray, right, and the metals, which are shown here in green and yellow and kind of burnt orange, and over here kind of in silver, okay? And then these are special elements. These are called the noble elements because they don't do any chemistry, all right? And they are group 8A, okay? And they're kind of special because they're found in their pure states in nature, which, according to the second law of thermodynamics, means that they're in a low energy state, meaning they're happy the way they are. So they don't do any chemistry. They just hang out, okay? What you'll notice with all the other elements is that they have different chemical properties than these guys in the last column because they have incomplete outer shell electrons, what we call valence electrons, all right? And so what guides all of chemistry is the effort on the part of all the rest of the elements to appear like these elements in this last column by either gaining, losing, or sharing electrons so that their outer shell is complete, which means that their outer shell is stable, okay? So, how do we use these different numbers that you see? Well, everybody in group 1A has 1 electron in the outer shell. Everybody in 2A has 2. Everybody in 3A has 3, and so on, okay? And then everybody in this row has a complete outer shell, meaning it doesn't need to make or break any chemical bonds. It just is made up of atoms of that pure element, okay? Now, with regard to chemical bonds, which are connections between atoms, okay? When metals react with metals, okay, so everything here to the left of the staircase over to the end here, they form what are called metallic bonds, which are things like alloys, and they're beyond the scope of this course, okay? When metals react with nonmetals, they form ionic bonds in which the metal loses electrons to the nonmetal, and that causes the atoms to take on electrical charges, okay? The metal becomes positively charged because it's lost a negatively charged electron, and the nonmetal becomes negatively charged because it's gained a negatively charged electron. And then what holds them together is the attraction of opposite charges, okay? And those are generally called salts, all right? When nonmetals react with each other, everybody here in silvery gray, to the right of the staircase and to the left of the orange pipe here, they form covalent bonds in which electrons are shared between the two atoms. But sometimes the sharing is perfectly equal, and sometimes the sharing is unequal, okay? The symbols are the letters here for the element. Right? There's boron, carbon, nitrogen, oxygen, fluorine, selenium. Over here, that's nickel, that's cobalt, right? There is, see if I could find, there's radium, there's hydrogen, lithium, sodium, and there is silver, there's gold, there's mercury. Okay? You'll notice the number at the top of the square is the atomic number, and it's always a nice round number, okay? And it is two things. It's the number of protons in the nucleus, but it's also equal to the number of electrons outside the nucleus. So if we take, for instance, carbon, okay? Carbon has atomic number 6, which means it has 6 protons in the nucleus and 6 electrons outside the nucleus, okay? The number on the bottom is called the mass number, and it's basically the number of protons and neutrons in the nucleus, but you'll see that it's not a nice round number. There are digits after the decimal point, and that's because when the universe was created, some of the atoms of each element were created heavy, meaning they had more neutrons in the nucleus than the most abundant, what we would call isotope of that element. And so what that means is if you were to go out and collect a certain number of those atoms of that element, and you were to weigh them, and then divide that weight by the number of atoms, what you would get is a weight a little higher than you would expect. Okay? The reason we care about isotopes in health science is because some isotopes give off a form of energy called electromagnetic radiation, which can be used in applications like X-rays. Okay, it can be used to destroy cancer cells in the body. It can be used to label molecules that we inject into your body and find out where they go. Okay, and that is a process that giving off of energy called radioactive decay. Okay, and when radioactive decay happens, sometimes that isotope will actually turn into another element because the particles in the nucleus will actually change. Okay, but when you're doing chemistry, which is just the making and breaking of chemical bonds, the identities of the elements do not change. Okay, but the chemical property of the molecule or the compound will be quite different from the chemical properties of what we call the reactants, which are the things that come together to make the molecule or the compound. Okay? So that's the periodic table. That's everything really you need to know with regard to the periodic table. We should point out, okay, that if we look at the structure of an atom, you can see, for instance, here with the carbon atom, right? Remember that carbon has the atomic number of 6, right? And there's a rule in the structure of the atom which states that the first orbital around the nucleus can hold a maximum of 2 electrons. The next orbital can hold a maximum of 8. The next orbital can hold 8, but is happy with up to 17, okay, and so on. All right. So when you're building your atom using the atomic number as a guide, the atomic number 6 for carbon means that the first 2 electrons will go in this inner shell and won't participate in any chemistry, and the remaining 4 will go into this outer shell, and they're called the valence electrons, and they will participate in the chemical bonding. Okay? And the reason for that is that the electrons in the outer shell have more energy associated with them that can be used for chemical bonding, whereas those that are closer to the nucleus are less likely to do that. Okay? So you can look at some examples of covalent bonds here. Hydrogen and carbon are considered non-metals when they react with each other, and they form covalent bonds which are going to result in the equal sharing, in this case, of pairs of electrons between hydrogen and carbon. Okay? And so that's a special kind of covalent bond that we call a nonpolar covalent bond. Whereas in water, we have the nonmetal oxygen and the nonmetal hydrogen sharing electrons, and we form what's called a polar covalent bond, okay, where the electrons spend more time with the oxygen than they do with the hydrogen. Okay. Notice in every case that in the outer shell, for the reactions that we're interested in, we have 8 electrons filling up the outer shell. You can see that here for oxygen, right? 2, 4, 6, 8 for the stable compound water. You can see that here in methane, 2, 4, 6, 8 for the stable compound methane. Okay, and then the hydrogens, of course, with only one shell, right, have 2 each in each of their outer shells. A full outer shell means a low energy state, which means that it's stable. So it's going to basically stay the way it is until something comes along to upset it in the form of energy. Okay, ionic bonds are different. Okay, covalent bonds are quite strong. Ionic bonds are electrical attractions between particles that have opposite charges. Okay, so what happens with sodium is that sodium with 1 electron in its outer shell, when it gets near chlorine, will punt this electron over to chlorine. The chlorine will take on a negative charge, the sodium will take on a positive charge, and the opposite charges will attract them to each other, and they'll form a salt crystal, which looks like a little cube. Okay? Notice that the positives are surrounded by negatives, and the negatives are surrounded by positives, and the reason for that is that that's a low energy state. Okay? And so that's the second law of thermodynamics again. All right? Now, with regard to these covalent bonds up here, that's an example of a polar covalent bond. This is a nonpolar covalent bond. Basically, with regard to interactions between molecules, similar chemical properties tend to associate with each other more easily. Okay, so nonpolar molecules tend to associate with nonpolar molecules, and polar or charged molecules tend to associate with each other. All right. An example of this is a bottle of Italian salad dressing. If you go and you take a bottle of Italian salad dressing off the shelf in the store, you look at it, you'll see that the oil, which is made up primarily of nonpolar organic compounds called lipids, okay, triglycerides, which we'll get into in a bit, are found on the top of the bottle in a layer, and then the salts and the water and the spices, which are either polar or charged, are found underneath. Okay, and that's the low energy state for that system. If you were to take that bottle and shake it up, you would form a temporary mixture called an emulsion. Okay, by adding energy to the system by you shaking it, right? But then when you put that bottle back down and wait, the oil and the rest of the material will separate again, adopting the low energy state, because that's what the second law of thermodynamics dictates. Okay. Now, with regard to ionic bonds, right? You know, for instance, that if you take a spoonful of salt and stir it into a glass of water, it'll dissolve, right? That's not news to you. I'm sure you've done that at some point, okay? The reason for this, though, is that the salt crystal is made up of negatively and positively charged particles. And when you throw it into water, the oxygen in the water has a slight negative charge because the electrons in the compound spend more time around oxygen than they do around hydrogen. And the hydrogens have a slight positive charge. So what happens is that the water molecules insert themselves between the chlorides and the sodiums, and then these ions just sort of fall away from the crystal and are surrounded by water molecules, and that's called solvation. Okay? So that's why you can dissolve ionic or polar compounds in water, and that's why water has the name the universal solvent. Okay? If you kept doing that, throwing salt into the water, eventually you get to a point where you throw the salt into the water and it wouldn't dissolve. It would fall to the bottom of the glass and stay there, okay? And that's called the point of saturation, right? And the reason that happens is because you've used up all the water molecules in the glass, solvating the salt that you threw in there previously, and there's none left to solvate the new salt that you just threw in. Okay? Polar covalent bonds in a single water molecule shown here, right? Hydrogen bonding is just a temporary attraction between the water molecules or between a, um, a compound that has a polar covalent bond in it that has a partially positively charged hydrogen, and in the other molecule a non-metal like nitrogen or oxygen in the case of water, okay, that has a slight negative charge. And so what happens is they stick to each other a little bit, okay, and what that causes is causes water to stick to itself, and that accounts for properties like surface tension and the fact that water expands upon freezing, okay? And it also accounts for water's high boiling point and high heat of vaporization and heat of fusion. Heat of vaporization is the amount of heat required to go from a liquid to a gas without changing temperature. And heat of fusion is the amount of heat given off when you go from a liquid to a solid and form ice, okay? So let's take a look at hydrogen bonding.
The atoms that make up a water molecule are in a constant tug-of-war over their shared electrons. Oxygen exerts a far stronger pull on the shared electrons than does hydrogen, and so the electrons spend more of their time closer to the oxygen atom. Because of this unequal sharing of electrons, the oxygen atom in a water molecule actually has a slight negative charge, and each hydrogen atom has a slight positive charge, even though the water molecule as a whole is neutral. Because of the unequal sharing of electrons and the resulting positive and negative poles, a water molecule is said to be polar. The polarity of water molecules causes them to be attracted to each other. Since the positively charged atom involved in this special type of attraction is always a hydrogen atom, this kind of bond between molecules is called a hydrogen bond. Each water molecule can hydrogen bond to 4 other water molecules. A hydrogen bond is weak and lasts only a tiny fraction of a second, but it takes a lot of energy to overcome the combined attraction of many hydrogen bonds. This explains water's great capacity to store heat, its high boiling point, surface tension, and several other unusual properties.
So, as we talked about earlier, right, like dissolves like. Polar things and charged things like to get together because it satisfies the second law of thermodynamics, okay? And nonpolar things like to get together for the same reason, okay? Similar chemical properties means they like to associate with each other because that means low energy, okay? And what you're looking at is just the interaction of a salt crystal and water, right? Form these little cages around, this is a solvation cage, and these things kind of float past each other, okay? An interesting thing, of course, about dissolving a salt in water is that now with ions in the water, the solution can conduct current, which is a form of energy, moving charged particles, okay? And there's a property we'll talk about a little bit later known as osmosis, which states that a solution of water and something dissolved in it will have the ability to pull pure water towards it, okay, because of a passive process that basically is the diffusion of water known as osmosis, okay. You can see here the water associating with itself through hydrogen bonding, the lipids or the nonpolar molecules associating with each other, okay, because they have similar chemical properties, all right? And basically, all that's going on here with like dissolving like is that when you have a positive and negative end to you, you can flip-flop those ends between molecules and get them to hug each other, right? Whereas with a nonpolar molecule, there really is no end to it. And so special forces, which we won't get into, called van der Waals forces, kind of cause those nonpolar compounds to get together and squeeze out the water between them, okay? And that all happens, again, without you having to do anything to the system, right? It's automatic. It is a passive process. Now, another very important property of solutions that we have to address is something called pH, okay? Now, the little p in pH has a definite definition, okay? The little p in pH, the little p stands for the opposite of the log to the base 10 of whatever comes after p, okay? So in the case of pH, it would be the opposite of the log to the base 10 of the concentration of protons, which usually has a designation with capital H and a plus after it, okay? So there's a property that always holds in water, all right? Very simple property, which is that the pH of a solution plus the pOH of a solution is always equal to 14. Okay. that's always true. All right. So that means if the pH of a solution is 6, the pOH of the solution is 8. If the pH of the solution is 7, the pOH is 7. If the pOH of the solution is 10, the pH is 4. Okay? So the reason this is important is because from pH, right, we can get an indication of the number of hydrogen ions there are in a solution. So if the pH of something, for instance, let's say is equal to 4 of a particular solution, right, then that's the same as saying that pH of 4, the proton concentration, the little brackets mean molar concentration of protons, okay, is equal to 10 to the minus 4th molar. Okay. Why do we care? All right. We care because organisms have a pH at which they operate best. All right. Where their life processes can proceed without damage. When you change that pH, you run the risk of creating toxic conditions that kill the organism. Okay, for instance, in a human, the pH of most of the areas inside the body, with just a few exceptions, is between 7.35 and 7.45. Okay, at that pH, the tools of life, which we're going to learn are called proteins, all right, are able to do their job because they can maintain their shape and carry out their function. And if we mess that pH up, then those tools will unravel and those functions will stop and the cell will die. Okay? And the same is true for a microbe, right? A microbe has tools of life, proteins, that operate best at a particular pH. Now, depending on the microbe, the pH is going to be different. Okay? Most microbes that cause human disease like a pH between 7.3 and 7.45 because they live inside us, right? But there are exceptions, right? Like Helicobacter pylori can basically stand a lower pH because the pH of the stomach is lower than the pH of, say, the blood, okay? But this is the reason we care, all right? Now, you might wonder what that capital M is, okay? That capital M just means molar, all right? And that just means the number of moles of something per liter. Moles per liter, okay? Liter is just a volume measurement. You've seen a 2-liter bottle of pop in the grocery store, okay? A mole is a count, okay? Like a dozen is 12, right? Or like a duo is 2, okay? A mole is 6.02 times 10 to the 23rd items, whatever we're talking about, right? So if we were talking about a mole of Buick LeSabres, that'd be a hell of a lot of Buick LeSabres, okay? But if we were talking about a mole of something like carbon, okay, we can go back to the periodic table here, and we can see that the atomic mass is 12.01. Well, it so happens that a mole of carbon weighs 12.01 grams, okay? So that's the usefulness of the mole concept, okay? When you weigh out 12.01 grams of carbon, you know exactly how many carbon atoms you have, okay? So don't confuse a mole with a mass. A mole is a count, okay? I'm not going to have you do any mole calculations or anything like that in this course. I just want you to understand what we mean when we say mole or molar, all right? and it was just developed as a way to be able to work with something on a bench as a chemist, um, in a quantity that you could easily manipulate and measure. Okay, so that's that's the bottom line there. Okay, that's all about pH and molarity. Okay, you can have a P next to anything, right? You can have a pH, you can have a pOH, you can have a pKa or pKb, pKsP. Okay, you can slap that P as a mathematical operator in front of any quantity, all right, and have a number come out the other end. And chemists do that a lot. Okay, we won't be doing that in this course, but I just wanted you to be aware of what that is. Basically, this is a measurement of the acidity. Okay, so the lower the pH, the more acidic the solution is. The higher the pH, the more basic the solution is. And neutral is defined as a pH of 7, which is the pH of pure water. Okay. Okay, we'll talk about organic molecules and lipids and carbohydrates. Okay, so organic molecules contain carbon and hydrogen in the same molecule. Okay, biomolecules are organic compounds that may include other elements like oxygen, nitrogen, sulfur, phosphorus. There's others that you'll find in there occasionally. Okay, the reason that they're called organic is because organic molecules are associated with life. All right. Now, why is that? Because carbon can do one thing that none of the other elements in the periodic table can do, which is it can form rings and chains with itself, which is really cool. Okay? Which means that with carbon, you can build anything you want using that sort of as a Lego piece. Okay? You've seen people play around with Legos, right? They have a few basic pieces, and depending on how you snap them together, you can build anything from a simple cube to the Taj Mahal or the Empire State Building. Okay? Um, so that's the neat thing about carbon. That's why it holds the place in life that it holds. Okay. If we look at some different organic compounds, okay, you can see here that these lines indicate covalent bonds, which are shared pairs of electrons between the two elements. Okay. You can see some examples here of some organic compounds. Ethane, right? 2 carbons and hydrogens bound to the rest. Again, if you do your bookkeeping, we note that the outer shell is full up, right? If each of these is 2, right? 2, 4, 6, 8 for carbon there, we're good, right? 2, 4, 6, 8 for carbon here, we're good. How about each hydrogen? 2 for that one, 2 for that one, 2 for that one, 2 for that one, 2 there, and 2 there, right? So we're good. It's a stable compound, okay? You can see some other, what they call functional groups in some other organic molecules, right? This is an amino group. It has a nitrogen in it, okay? This is a carboxylic acid group, okay? It has a carbon double bound to an oxygen bound to an OH in it, okay? Acids tend to be proton donors, meaning they lose their hydrogen on their group and they cause the pH of that solution to go down, all right? Whereas a base is what they call a proton acceptor, which means it scoops protons up from the solution and causes the pH to go up, okay? You can see some other functional groups over here. The ester group, it's an example of an ester called methylformate, right? There's ethanol, which you drink in beer and wine. There's dihydroxyacetone, which is an intermediate in a process we're going to learn about known as glycolysis, okay, which is inside every living cell in the body, pretty much. Okay. Now, an important thing to understand about these pictures is that they don't quite give you the whole story, right? To look at these drawings, you would think that all these molecules are flat, and they're actually not flat, okay? The carbon atoms that are attached to 4 other groups, they actually have a shape known as tetrahedral, okay? And a tetrahedron basically looks like a carbon atom with sort of a 3-legged stool, where this would be one leg of the stool coming out at you, okay? This would be a leg of the stool going into the board. Okay. Daggone it. Switch. Switch. There we go. Going into the board. Okay. Color black. There we go. So this is going into the board. Okay. And then this would be a leg that's in the plane of the board. Okay. And then there would be another leg up here that's in the plane of the board. Okay. And we could put, if this is methane, hydrogens at each of these. Okay. So this thing has a shape to it. Right. It has a three-dimensional shape. It's not just flat like a pancake. Right. And the reason I bring that up is because when we start talking about proteins and stuff, you're going to want to understand that every organic molecule is going to have a shape associated with it, and those shapes are important because they're going to be a factor in how the things interact with each other. Just like a key fits into one particular lock, right? Molecules with certain shapes like to hug each other, okay? So just be aware that that's the case. Now, when a carbon is attached to three other groups, such as here, it's flat like a pancake. And when a carbon is attached to two other groups, like in a molecule called carbon dioxide, it is in a straight line. It's called linear. Okay, so just keep that in mind. Now, the reason for these shapes is, again, the second law of thermodynamics, which just states that we want to have the lowest energy arrangement we possibly can, and that carbon atom attached to those other groups, we want to keep those other groups as far away from each other as possible because they have electrons around them, and we don't want those electrons pushing on each other more than need be. Okay, and so those are the shapes that get that done the easiest. Okay, all right. A lot of biomolecules are polymers, which means there are chains of repeating chemical units. Examples include lipids, such as triglycerides, steroids, and phospholipids. A component of a triglyceride is something called a fatty acid. Another component is a sugar called a glycerol. Okay, carbohydrates, which come in many forms: monosaccharides, disaccharides, polysaccharides. We'll learn about those. Nucleic acids, which are information molecules built from building blocks called nucleotides, and proteins, which are built from building blocks called amino acids. Okay, so our first topic are lipids. Okay, lipids get their name from the fact that they're mostly nonpolar. Okay, meaning they're greasy or oily. All right, and so they like to be with each other. Okay, the components of triglycerides are fatty acids and glycerol. Well, what's a fatty acid? There's an example of a fatty acid, right? That's in the acid group of the fatty acid. That actually has some charge to it, okay? This is the greasy part down here, all right? Because there are no atoms in this chain that are to the right of carbon and to the left of the noble elements in the periodic table, okay? So there's no nitrogens, no sulfurs, no oxygens anywhere in here, all right? It's just carbon and hydrogen. So when you see carbon and hydrogen being most of the structure of the molecule, you can say that it's nonpolar, basically, okay? Fatty acids condense with glycerol to form triglycerides. We'll see that in a minute, where this end of the molecule has a little bit of polar character to it, and this end is greasy. And the result is that we call this an amphiphatic molecule. Okay, amphiphatic. Let's see here. Let's see. Oh yeah. Okay, got it. So an amphiphatic molecule. Okay, it should be AMPH AITAC. Amphiphatic. That's not doing it for me. Take this down. Okay. Has a part that is nonpolar and a part that's polar. Okay, and in water, what will happen is that the polar part will be associated with the water, and the greasy part will get with the other greasy parts. Okay, so if you throw a triglyceride into water, what it'll do is probably form a little bubble where the fatty acid tails project inside, and the part that has triglyceride on it is on the outside next to the water. And a phospholipid, we're going to learn, is a membrane component, and it has an even more polar part because it has a phosphate group in place of one of the fatty acids, and so it has an even more polar to charge character, which makes it an amphipathic molecule as well. Okay. Important thing about lipids is that they have properties that make them useful for cell membranes, which we'll learn about. And they also have more calories than any other molecule that can be available as fuel. And the reason for that is they have more carbon-hydrogen bonds than either a carbohydrate or a protein. Okay? And the carbon-hydrogen bond is the most energetic bond for living organisms, right? When we crack that chemical bond, more energy comes out that can be used to do work. Okay? So let's take a look at lipids.
Lipids are made of carbon, hydrogen, and oxygen atoms, arranged in long chains consisting mainly of carbon-hydrogen units. 3 fatty acid chains can be linked to a glycerol molecule to produce the lipid known as a triglyceride. As usual, the fact that a dehydration synthesis takes place tells you that the reverse reaction, hydrolysis, is also possible. Phospholipids are one very important class of lipid. Phospholipids make up the plasma membrane of the cell. Their function is directly tied to their structure. The head of the phospholipid is hydrophilic, which means it is attracted to water, while its tail is hydrophobic, which means it is repelled by water. When phospholipids are surrounded by water, they naturally form 2 layers so that the hydrophilic heads are always turned out toward water, and the hydrophobic tails are always turned in toward one another, where the water cannot touch them.
Butter, lard, margarine, and salad oil are composed of lipids called fats. To make a fat molecule, 3 fatty acids bind to a molecule of glycerol. For this reason, fat molecules are technically called triglycerides. Fat molecules do not mix with water because they have 3 long hydrocarbon tails. Cells use fats for energy storage because the tails hold more potential energy than other biological molecules.
Now, carbohydrate is a little bit different. Okay, carbohydrates always have the chemical formula CH2O, and then we put that in parentheses, and we put a little N down here to represent the number of carbons in the molecule, right? So if I have, for instance, a 6-carbon carbohydrate, the formula would be C6H12O6. Okay? Now, because of the large number of electronegative atoms, there's a lot of oxygens in a carbohydrate, it is polar, which means it dissolves easily in water. And y'all know this anyway. You know you can dissolve sugar in water. That's not news to you. Okay? Carbohydrates in living systems tend to have rings in the structure which either have 6 or 5 sides, okay? But they can also exist as linear molecules, and they can kind of flip back and forth between those 2 shapes, all right? Carbohydrates are used primarily for energy, but they're also used to build cell walls. They are used as identification molecules on the surface of cells known as antigens. Okay? They are part of the structure of RNA and DNA, nucleic acids. Okay? But their big use is an energy storehouse. Okay? This is not storehouse, but really just energy fuel. Okay? The reason, there's a lot of carbon-hydrogen bonds in carbohydrates. Okay, not as many as in a fat, but still quite a bit. Okay, you can see here some examples, right, of going from monosaccharides to disaccharides. Di meaning two, right? Two rings here. There's one, there's another. This is through what's called a dehydration synthesis reaction, in which a water molecule is removed using parts of both of these sugars, and that forms a brand new linkage called a glycosidic bond between the two rings. Okay? And that is a chemical reaction that does not happen on its own, so it has to borrow energy from another source. All right? And we'll get to what that other source is in a bit. This also falls under what we would call an anabolic reaction, in which you need energy from some other place for the reaction to happen, all right? As opposed to a catabolic reaction in which we would go in the opposite direction. We might go from, say, a disaccharide to two monosaccharides, and in the course of doing that, we would take up a water molecule, okay? Instead of kicking it out, we would take it up because we would use the water molecule to bust this bond, and that would release energy. And that's called a catabolic reaction. So the energy that comes out of catabolic reactions is used to drive anabolic reactions. Okay? And the combination of all the chemical reactions in an organism is called its metabolism. Okay? So let's take a look at carbohydrates.
Let's talk about the structure of carbohydrates. You've probably heard that carbohydrates are considered either simple or complex. But what does that mean? A carbohydrate is simple if it contains only 1 or 2 units of sugar. We use the term monosaccharide when discussing carbohydrates with only 1 unit of sugar, and we use the term disaccharide when discussing carbohydrates with 2 units of sugar. A carbohydrate is complex if it has many units of sugar linked together. The term we use to describe that is polysaccharide. Here are the 3 monosaccharides that you may find in your diet. They are glucose, which is the sugar in your blood right now, fructose, which is the primary sugar in fruit and other plants, and galactose, which is part of milk sugar. See how each of these are single sugar units? Here are the disaccharides found in food. They are maltose, or malt sugar, sucrose, which is what you know as table sugar, and lactose, which is milk sugar. Notice that these disaccharides are made up of 2 monosaccharides linked together. Next, we have the complex carbohydrates called polysaccharides. These are chains of many monosaccharides linked together. Examples of polysaccharides are starch, glycogen, and fiber. Starch is the storage form of glucose in plants. The plant is able to use sunlight and carbon dioxide and water to make glucose. And then it can stockpile those glucose molecules by forming long branched chains of glucose called starch. A potato is a very visual example of this. The potato plant has stockpiled many glucose molecules into starch in this potato. It can rely on the stored glucose when it needs energy, such as when there isn't much sunlight available. Another polysaccharide is glycogen. Glycogen is much like starch in that it is chains of glucose molecules linked together. But glycogen is found in humans and other animals, not plants. You stockpile glucose in your liver and your muscle cells, just like the potato stockpiles glucose as starch. You use glycogen for a similar function. When you need glucose, you can mobilize it from your glycogen stores. The final complex carbohydrate we will discuss here is plant fiber. Plant fiber is composed of glucose molecules linked together, just like starch and just like glycogen. But the glucose molecules are attached by bonds that cannot be broken down by human or animal enzymes. Because of this, we cannot digest plant fiber, which means we cannot absorb the glucose molecules into our bloodstream. Instead, they pass through our digestive tract, doing some fascinating things to improve our health.
Carbohydrates are made of carbon, hydrogen, and oxygen atoms arranged in various ring structures. The disaccharide sucrose is formed in a dehydration synthesis involving the monosaccharides glucose and fructose. When two monomers combine to form a dimer, a water molecule is produced from the dehydration synthesis reaction. Notice which parts of the two monomers were involved in forming the sucrose. The glucose, OH, that gave up its H is the one on the carbon next to the ring oxygen atom when you move clockwise from the oxygen atom. The OH lost from fructose is on the carbon next to the ring oxygen atom when you move counterclockwise from the oxygen atom. When a complex carbohydrate breaks down to simple sugars via hydrolysis, a water molecule is split between the two polymer subunits.
Polysaccharides are polymers, long chains consisting of hundreds to thousands of linked monosaccharides.
Okay, now we'll talk about the information molecules, DNA and RNA. We'll talk about nucleotides as well. Okay, you're going to find out that DNA and RNA are basically information molecules, right? The instructions to build the proteins that do the work of life are contained in DNA. They are then transferred, those instructions, to RNA, and then the instructions in the RNA are used to build the proteins, okay? The movement of the information from DNA to RNA is called transcription, and the movement of the information from the RNA to the protein is called translation, okay? In bacteria, transcription and translation happen in the cytoplasm at the same time, near the nucleoid region, and in eukaryotes, transcription happens in the nucleus, and translation happens in the cytoplasm. Okay? So let's take a look at DNA and RNA.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The two DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The two polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
Okay, when we look at RNA, we're looking at a molecule that's involved in the process of translation. There are different flavors of RNA. Messenger RNA is transcribed from a gene in the DNA and contains protein building information. Ribosomal RNA is a structural component of the ribosome, and transfer RNA is used to bring amino acids to the ribosome and the messenger RNA. Now you might be wondering, what's a ribosome? A ribosome is basically an organelle in the cell. It's a combination of protein and RNA that makes protein using the instructions in a messenger RNA. So it reads the message just like reading a recipe in a cookbook, and then it goes about the job of hooking the amino acids together that are going to make the protein. Okay? Now, it's true that in some microbes, okay, the genetic information, for instance, in viruses, can be RNA instead of DNA. Okay, viruses are unusual in that they're not technically living things, and they can have genetic information that can be, depending on the type of virus, either double or single-stranded DNA or double or single-stranded RNA. Okay. So that's a little bit unusual. Now our next topic is proteins. The building blocks of proteins are called amino acids. Okay, and you can see the different amino acids here. All right, and the amino acids all have a common chemical structure in their backbone. Okay, this is the same in every amino acid. Right, and what differs from amino acid to amino acid is what we call the R group, which can be a collection of different atoms depending on the type of amino acid that it is. Okay, and we can divide amino acids into charged, polar, and hydrophobic or nonpolar amino acids. Okay, meaning what? Meaning that the R groups in the nonpolar group don't really have much separation of charge on the R group. The polar amino acids have electronegative atoms in them and some separation of charge. And the charged amino acids have an outright charge on their R groups. Okay? So let's look at protein structure.
Proteins are polymers of amino acid monomers. Every amino acid contains a central carbon atom bound to 1. An amino group, 2. A carboxylic acid group, 3. A hydrogen atom, and 4. A group that differs from one amino acid to another and is represented by the letter R. This dehydration synthesis reaction has created a protein consisting of 4 amino acid monomers.
Each protein has a unique primary structure, a particular number and sequence of amino acids making up the polypeptide chain. 20 different amino acids are used to build proteins. Theoretically, the various amino acids could be linked in almost any sequence, forming an almost infinite variety of different proteins. This illustration shows some of the amino acids making up the primary structure of a protein. The structure of a single generalized amino acid is shown below. The main backbone of every amino acid is the same. This is what forms the backbone of the polypeptide chain. It is the R group, which projects out from the backbone, that makes each of the 20 kinds of amino acids unique. Different amino acids have different properties that affect the folding of a protein. Thus, primary structure ultimately determines the shape of a protein, which determines its function. In most proteins, parts of the polypeptide chain are coiled or folded, forming twists and corrugations. This is secondary structure. The turns and folds of secondary structure contribute to the protein's overall shape. One kind of secondary structure is the alpha helix, where the chain twists. Another is the pleated sheet, where the chain folds back on itself, or where two regions of the chain lie parallel to one another. Secondary structure results from hydrogen bonding between atoms along the polypeptide backbone. Oxygen and nitrogen atoms along the backbone are highly electronegative, giving them partial negative charges and leaving nearby hydrogen atoms with partial positive charges. These negatively and positively charged atoms are attracted to one another at regular intervals along the chain, causing parts of the protein to twist or fold back upon itself. Superimposed on primary and secondary structure is tertiary structure, irregular loops and folds that give the protein its overall three-dimensional shape. The irregular folding of tertiary structure results from interactions among the R groups of amino acids. Acidic and basic R groups ionize, and these positively and negatively charged groups may form ionic bonds. Polar forces also contribute to tertiary structure. Hydrophilic or polar R groups may hydrogen bond with one another or turn outward and hydrogen bond with the surrounding water. Hydrophobic, nonpolar R groups cluster on the inside of the protein away from water. Tertiary structure may be further stabilized by strong covalent bonds between sulfur atoms in certain R groups. Some proteins consist of two or more polypeptide chains. The fourth level of protein structure, quaternary structure, results from the combination of two or more polypeptide subunits. Quaternary structure is stabilized by the same sorts of attractions that stabilize tertiary structure. Hemoglobin, the red oxygen-carrying protein of blood, is an example of a protein with quaternary structure. It consists of two kinds of polypeptide chains. Two of each, a total of 4 chains, make up each hemoglobin molecule.
So, what's important to take home about protein structure is that once the primary structure of the protein is complete, and that's done by the ribosome using the information in the messenger RNA, then the protein will begin to adopt what's called its native conformation, meaning the shape in which it can do its job properly. And what drives that change in shape is the association of polar R groups with polar R groups, charged R groups with R groups of the opposite charge, and nonpolar R groups with nonpolar R groups. Okay? And that guides the formation of first the secondary, then the tertiary, and then, if it's present, the quaternary structure. And once that happens, okay, that protein is generally ready for business. In some cases, for instance in eukaryotes, there may be some chemical modification that goes on before it's ready really to do its job. We might add carbohydrates or lipids to some of the R groups, for instance, or phosphate groups, okay? But it depends on the protein, and this is generally something that's happening in eukaryotes and not prokaryotes, okay? And anything that interferes with that native conformation is going to ruin that protein's tertiary, quaternary, and secondary structure and cause it not to be functional, and that's a process known as denaturation, okay? And factors that can do that include changes in temperature, changes in pH, and changes in salt concentration, as well as mechanical force, okay? So the organism will always try to exist in an environment, both externally and internally, that allows its tools of life, its proteins, to work properly, okay? And different organisms like different sets of circumstances, right? We're going to learn about some organisms that are perfectly happy in the human body in places like the bloodstream. We're going to learn about some organisms that prefer an environment that is the equivalent of boiling battery acid, okay? And the reason that those organisms can exist in those different sets of circumstances is because they have different tools of life. Now, how did this happen? How did we get such different tools so that these organisms could carry out such a different lifestyle? Well, that's a process that you've learned about in biology called evolution and natural selection, in which a population of organisms has differences in their genetic material, which lead to differences in their proteins, which lead to differences in their characteristics, okay? And in a certain environment, certain characteristics will be favored, meaning those organisms will live long enough to breed, and then there'll be other organisms that won't be favored, okay? And the result is that we will select for characteristics that make it more likely that that organism will survive and pass its characteristics on to its offspring through its genetic material, okay? And over time, the collection of these changes, which comes from a process known as mutation, which are changes in DNA or in some cases RNA, will lead to a new species, okay? And so that's how we get the variety of life we see on the planet today, evolution and natural selection. You can see natural selection in real time. You can observe it. In fact, we will observe it, okay, in some of the E-labs we're going to do. And then evolution is something that you can't observe in real time because it takes many generations, okay, but the evidence for it is found primarily in the fossil record. Okay, we'll talk about how energy and entropy determine the direction of reactions, and we'll talk about oxidation and reduction reactions, and talk about how enzymes catalyze reactions. So we talked a little bit earlier about the fact that organic molecules are going to have energy in them, in their chemical bonds, okay? And that the money bond for living systems is the carbon-hydrogen bond. There's more energy in that than there is in any other bond in a fuel molecule that's taken up by an organism, okay? So when you crack that bond, energy comes out, but that energy can't be directly used to do work yet. It has to be transferred to a molecule called ATP, adenosine triphosphate, which is a nucleotide. It is one of the building blocks of RNA, okay? But in this case, it serves as readily available energy, or I like to call it biological gas in the tank, okay? The cell just isn't set up to directly use the energy from lipids, proteins, and carbohydrates to do the work, okay? It needs to go through this extra step in order to turn into something that is good to go, okay? An analogy would be your car, okay? Let's say you got your car, it's sitting at the bottom of the hill, okay? And the energy that makes your car go comes from gasoline that you get at the gas station, okay? And what happens when you turn the ignition and push down on the accelerator pedal is that gasoline gets mixed with oxygen in the air in what's called the carburetor, and then sprayed into an area of the engine block where spark plugs and pistons are located. The spark plug makes a spark, the vaporized gasoline explodes, and that moves the pistons in the engine block up and down, and that turns the crankshaft, and that eventually turns the wheels of your car, okay? And your car goes uphill. And that's an example of coupling a favored chemical reaction to an unfavored process, right? Your car isn't just going to automatically roll uphill. The second law of thermodynamics says that's a no-go, okay? But when you crack the bonds in gasoline, you release energy in the form of pressure, heat, and light, right? And then we can use that to do the work of getting the car to roll uphill, and we'll give off some heat in the process. And that heat will go off and make the universe more random. And so that's what's called increasing the entropy, okay? Well, where does that gasoline come from? Okay, that gasoline comes from crude oil, which is a black viscous liquid that comes out of the ground, okay? But if we were to take crude oil and put it in your gas tank, your car wouldn't run. And the reason is that your carburetor and your engine block and your spark plugs and everything don't understand the energy that's contained in crude oil. They can't use it easily, okay? So to get the energy out, what we do to crude oil is we crack it. So we crack it into tinier pieces. Crude oil is basically long hydrocarbon molecules that are hydrophobic. And the piece that's used in gasoline is called octane. Now, you've probably heard of an octopus with 8 arms. Octane has 8 carbons, okay? And so it's the high octane fuel at the gas pump that your car engine can understand and use to turn into what we call kinetic energy, which is the energy of movement. So we go from chemical potential energy, the energy stored in the chemical bonds, to kinetic energy, which is the energy of movement. Well, the same deal is going on in the cell, right? We got usable energy, right, that we need to steal from the fuel we take in, and then we need to turn it into both usable energy that is available to do work in the form of ATP, and then we're going to give off some as heat, okay? Now, when we talk about chemical reactions, okay, one of the things that happens in chemical reactions is that we have a certain amount of energy contained in what are called the reactants, which are the things that are going to get together or fly apart to make what are called the products, okay? There's a certain amount of energy in the reactants, there's a certain amount of energy in the products, okay? If there is more energy in the reactants than there are in the products, then the reaction is said to be spontaneous, meaning it'll happen by itself. It'll just go, okay? If there's more energy in the product than there is in the reactant, then the reaction is not spontaneous. And so in order for it to go, it needs to steal energy, usually from another chemical reaction, okay? We gave the example of your car going uphill, right? The energy to make that happen came from the exploding gas in your engine block, right? That was a favored reaction, okay? And the unfavored sequence of events was the car rolling uphill, okay? Well, in the body, all right, unfavored reactions are things like building proteins or building RNA and DNA and polysaccharides and cell components, right? Those things don't just assemble themselves automatically. They have to draw power from some other chemical reaction, and that reaction is the hydrolysis of a molecule called ATP, all right? Well, to make ATP, we have to steal the energy from the fuel, and that happens through what are called oxidation reduction reactions, in which we take highly reduced molecules, ones with lots of carbon-hydrogen bonds, okay? And then we oxidize them, so we turn them into compounds with fewer carbon-hydrogen bonds, right? And then we send those electrons that we steal from that reduced compound, usually to a carrier, right? that is a molecule that holds those electrons temporarily, and then eventually we take the energy from those electrons and use that to make ATP, okay, in a process known as metabolism. All right, so there are different sets of reactions that make up metabolism. In eukaryotes, metabolism is made up of a series of reactions called glycolysis, where we take in glucose and we produce something called pyruvate, as well as something called NADH. Okay, and then a second set of reactions called the Krebs cycle, in which we take pyruvate in and we produce CO2 and water and NADH and something called FADH2, right? And then finally something called the electron transport chain, in which we take in NADH and FADH2 and ADP and phosphate, and we make lots and lots of ATP, okay? Then there are microbes that have variations on that as well, okay? Biochemical reactions are aided by proteins that are called enzymes, which come into chemical reactions to make them happen more effectively without having to change the concentration of the reactants or the temperature of the reaction, okay, which would create conditions that would kill the organism, right? So you could think of the enzyme as like a tool that helps the chemical reaction happen super quick, right? And that particular tool, that enzyme, has properties known as a catalyst. Okay, a catalyst is something that participates in a chemical reaction without getting consumed by it, right? So let's take a look at what enzymes do and how they do it.
For any reaction to occur, even one that releases energy, some energy must be added to get the reaction going. This energy is needed to break bonds in the reactant molecules. The energy needed to start a chemical reaction is called the energy of activation, EA. This required energy input represents a barrier that prevents even energy-releasing reactions from occurring without some added energy. How does a living cell overcome the energy barrier so that its metabolic reactions can occur quickly and precisely? A special kind of protein called an enzyme is the answer. An enzyme serves as a biological catalyst, increasing the rate of a reaction without being changed into a different molecule. An enzyme does not add energy to a reaction. Instead, it speeds up a reaction by lowering the energy barrier. An enzyme is very selective. Its three-dimensional shape allows it to act only on specific molecules, referred to as the enzyme's substrates. As the substrates bind to the enzyme's active site, they are held in a position that facilitates the reaction. This takes less activation energy than the unaided reaction. Products form and are released. The enzyme emerges unchanged from the reaction. Because of the specific fit between enzyme and substrate, each enzyme can catalyze only one kind of reaction involving specific substrates. Thousands of different enzymes may be required to carry out all of a cell's metabolic processes.
Okay, we're going to talk about cell membranes next, talk about what they're made of, talk about what the cell membrane does for the cell, and talk about membrane transport as well. Okay, in a living organism, right, the cells that make up that organism have a barrier known as the plasma membrane. All right, the main building block of the plasma membrane are molecules called phospholipids, and we already know from our previous discussion that phospholipids have a greasy part and a polar to charged part, right? So in water, what a phospholipid, an amphipathic molecule, will do is have the polar or charged portion interact with either the water outside the organism or the cytoplasm inside the organism, right? Because that's what the second law of thermodynamics says is going to happen, okay? And then the greasy parts, the fatty acid tails, will form what we call a phospholipid bilayer, right? Where the greasy part is in the middle and the charged or polar parts are on the edges, all right? And that forms a semi-permeable barrier, which is one in which only certain molecules can go through. Okay, so the kind of things that can pass through a phospholipid bilayer are anything that's non-polar, okay, such as oxygen molecules or things like steroids, which are another type of lipid, okay. Certain gases can pass through, like nitrogen gas, okay. And water can go through primarily because of its small size, okay. And that's sort of an exception to the rule because water, as we know, is polar, right? But the membrane itself is actually not just made of phospholipid, right? It also has carbohydrate on it. It has protein embedded in it or on either side of the membrane. The proteins that are on either side are called peripheral proteins. The proteins that go all the way through are called integral proteins, all right? The purpose of the integral proteins is usually to serve as a gate, a channel, or a pump, okay, or sometimes a receptor, okay? And the purpose of the peripheral proteins is usually to set off a series of chemical reactions that lets the cell respond to the changes in the environment outside, or to serve as ways to anchor a component of the cell called the cytoskeleton to the membrane, and then in the case of tissues to neighboring cells, okay? So that's what's called a fluid mosaic model, right? It's not just one thing, it's several things, okay? So let's take a look at some different aspects of the cell membrane and membrane transport.
Like the walls of a house or building separate the interior from the outside world, the cell membrane is a barrier that separates the contents of the cell from the external environment. Cell membranes are a mosaic of protein and lipid molecules, both of which can drift from place to place within the membrane. Most of the surface area of the membrane consists of a type of lipid called phospholipid. Each phospholipid molecule has a polar head and a nonpolar tail. The phosphate-containing head is hydrophilic, or water-loving, because its polar structure is attracted to water molecules, which are also polar. The fatty acid tail is hydrophobic because it is nonpolar and repels water molecules. Cell membranes are phospholipid bilayers, a double-layered structure in which the hydrophilic heads of the phospholipids face outward. The middle of the membrane contains the hydrophobic tails of the phospholipids, an arrangement that shields them from contact with water molecules. Whereas membrane lipids act as a general barrier around the cell, membrane proteins have specific functions. For example, they may act as enzymes, receptors, anchors, or channels. There are two major populations of membrane proteins: integral proteins, which are part of the membrane structure and usually span the width of both phospholipid layers, and peripheral proteins, which are bound to the surfaces of the membrane.
Molecules dissolved in a solution are in constant random motion due to their kinetic energy. One result of this motion is that dissolved molecules become evenly distributed throughout the solution. This tendency of molecules to spread out is an example of diffusion. But how do these molecules come to be evenly distributed? Let's start with a beaker of plain water. What will happen if we now add a lump of sugar to the water? A lump of sugar is composed of many individual sugar molecules, and even as a solid lump, the individual sugar molecules are in motion. When the lump is dropped into the water, it begins to dissolve. Individual sugar molecules move randomly and constantly from the area where they are common to the area where they are scarce. This type of motion, when molecules move from areas of their higher concentration to areas of their lower concentration, is called diffusion. Diffusion continues until all the sugar molecules become evenly dispersed throughout the beaker. The rate of diffusion is affected by temperature, size of molecules, and the steepness of the concentration gradient. Although not specifically shown in this animation, this is one of the processes whereby materials are exchanged between a cell and its environment. Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
The image on the left is human erythrocytes, or red blood cells. The image on the right is enhanced with filters. These living cells are in isotonic solution. Note the lozenge shape. Now the solution has been changed to have a high salt concentration. It is hypertonic. The red blood cells shrivel and become crenated. Finally, the solution has been changed to plain water. The cells swell and burst, leaving empty membranes behind. This phenomenon is called hemolysis.
Okay, we'll talk about vesicle transport and talk about how pathogens take advantage of vesicle transport. Okay. There are three types of vesicle transport, really more than three types, but three major types: endocytosis, pinocytosis, phagocytosis, and then sort of a blanket term that covers both pino and phagocytosis called, an opposite of that process, which is called exocytosis. Okay, so in endocytosis, what happens is that the cell membrane will invaginate and pull some component in from the outside, and that'll form what we call a vesicle, and then that vesicle will have a variety of different possible fates, right? That vesicle might end up fusing to another organelle in the cell called a lysosome, right? Which is basically where powerful digestive enzymes are located, and then we break down what's in that vesicle and then use some of the pieces, and some of the pieces we get rid of, okay? Or we may, through endocytosis, carry that material to another organelle than a lysosome and use that in some other chemical process, okay? In pinocytosis, we're usually taking in liquid. In phagocytosis, we're taking in solids, right? And in receptor-mediated endocytosis, we're taking in a particular ligand, okay? So there are, in some cases, receptors on the cell membrane that that bind a particular chemical, and then that receptor-mediated attachment of the chemical to the receptor triggers the production of a vesicle that then travels around inside the cell to a particular location. Okay? Exocytosis is the opposite. In exocytosis, the cell manufactures something, puts it in a vesicle, and then spits it out. Okay? All of these are active transport processes. They need ATP to go because they don't happen spontaneously. right? So let's take a look at endocytosis and exocytosis.
The substances used as fuel by single-celled organisms include other smaller cells, particles of organic material, and large molecules that cannot pass through the plasma membrane. Many single-celled eukaryotes use a mechanism called endocytosis to ingest such food particles. In this process, the plasma membrane surrounds and engulfs the food particle. Cells use three basic types of endocytosis depending on the size and nature of the material to be ingested: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterial cell or an organic fragment, the process is called phagocytosis. If the material is a liquid, it is called pinocytosis. Some types of molecules, such as low-density lipoproteins, LDLs, are transported across the plasma membrane by receptor-mediated endocytosis. These molecules first bind to specific receptors embedded in the plasma membrane. The receptor molecules are concentrated in an indented pit coated by the protein clathrin. When sufficient target molecules accumulate in the coated pit, the pit deepens, seals, and is incorporated into the cell as a coated vesicle. Exocytosis is the reverse of endocytosis. This process results in the discharge of materials from membrane-bound packages that migrate to the inner surface of the plasma membrane, fuse with the membrane, and then release their contents to the outside of the cell.
Okay, these are just some links to some useful animations that add to what we've already learned. I will embed these in the PDF that comes with this. So the link will be here with the little figure, and you can go and watch each of these. One is on vesicle transport. One reinforces osmosis and water balance in the cell, and the other is on oxidation reduction reactions. Okay, so make sure and take a look at those in addition to what you've already seen in the podcast. And I thank you for listening.